Hello, I'm Jo Fay, and this is a SwissInfo podcast. SwissInfo is the multilingual and international public media company of Switzerland. Today we're off to one of the biggest glaciers in the Alps. Like many others, the Rhone glacier is melting. And as it melts, it releases ancient microorganisms that have been living in the ice for thousands of years. Now it's time for scientists to find out what these organisms look like. The Rhone glacier stretches across the eastern end of Canton Valais in southern Switzerland. It's where the water of the River Rhone comes from, one of the primary sources of Lake Geneva. The glacier is melting rapidly, and it's estimated it'll be gone by the end of the century. The melting water is of special interest to a group of scientists. They're from the Federal Research Institute for Forest, Snow and Landscape, the WSL. They spent a lot of time working on the Rhone glacier this summer. Our journalists, Celine Stegmüller and Luigi Iorio, went to have a look. Here's Celine.
It was at the end of August when we rented out 2 pairs of crampons from a local shop and made our way up the Rhone glacier. We joined a group of biologists from the WSL. They had been collecting microorganisms, viruses and fungi from the meltwater for weeks using special pumps, plastic bags and filtering systems. Team leader Beat Frey told us what was driving them.
We go everywhere, we go to the moon, but still we even don't know what we have here on the ground. So what is living on the ground? So what bacteria, what fungi? And they actually are fundamental for our life to surviving. And many of them, they're unknown. So we know which birds we have, how many birds, how many insects we have above ground. We can protect them. We have rare species, but from the soil we don't know, or from the ice or from permafrost we don't know anything about why they have a huge diversity. I mean, there are thousands of different species underground, and we don't know what they are doing. And I think this is very fascinating to go into this treasure.
Frey told us when they started the project, the researchers had to figure out how to collect the microorganisms.
So either we take ice directly to the lab, hundreds of kilos of ice, it's not easy to bring them into the lab. So then we had the idea, we could directly filter the meltwater on the glacier surface. So either in ponds, which feed from underground melting ice, or from glacier surface streams.
And that's exactly what they were doing while we spoke to Frey. One group worked on the meltwater inside the glacier, while the other one filtered and collected meltwater on the surface. The gear is all custom made.
So we constructed a filter apparatus. We constructed a system that we can filter water, and that the particles, mostly bacteria, fungi and some particles, are absorbed on the filter. Hundreds of liters we can filter here, and then we have all on a filter, and we extract then the DNA, and we know what bacteria, what fungi, what viruses, what algae.
The scientists collect water from bright blue crevasses as well as from small streams running through the ice. They do that with a special nozzle, a thing that looks a bit like a shower head. The nozzle can be opened to insert sheets of filter. It's then plunged into the water, which is pumped through tubes and into big plastic bags. Larger microorganisms will stay on the filter. To get the smaller microorganisms, as well as the viruses, it requires an extra step. The researchers are adding iron particles to the water inside the plastic bags. The smaller organisms and viruses will now bind to the iron particles. The water is pumped through the filter a second time. This will also hold back the smaller microorganisms and viruses. But why are the researchers interested in these microbes?
They have to tackle very harsh conditions in relation to sunlight, to energy, to nutrition, to water, temperature. So that we get out microorganisms, special features, genomic features, and this we would like to know a bit more about and hope that we, from this information, we get some organisms which have very special features which potentially can be used in industrial applications.
For instance?
For instance, antibiotics production, so that they produce kind of special antibiotics. Another interesting feature is that they use special polymer degradation, so plastic degradation, because they have very special enzymes or proteins which can degrade the plastic. So that's the other hope, and we have already nice candidates on plastic degradation from this kind of environments. But for this it needs that we bring them into the lab and cultivate them there. So that's the other big challenge, because we don't know the conditions where they live in actually.
So we may think in the ice, there's no life in the ice, in the glacier, so harsh conditions. So do you know what are you looking for?
We know for most of the organisms we know what we expect, most, but maybe 40% we don't know. So this is a bit scary in a way that we don't know with whose organism we are working on. So therefore that's also a challenge a little bit then to work with some unknowns, and of course we have to make some precautions in the lab also that we work properly under sterile conditions.
Frey told us that some of these organisms could also be harmful to current microorganisms. That's another reason to find out more about them. 2 months after our visit to the glacier, we joined the team in the lab. This was at the headquarters of the WSL Research Institute near Zurich. The filter sheets with the bacteria, viruses and fungi collected on the Rhone glacier are kept in a fridge waiting to be processed. Biotechnologist and scientific assistant Benedikt Grunz, who was also on the glacier with us, showed us around the lab.
So this is the lab where we work in. So we come from the glacier, bring the filters here and try to keep it all cooled, like always at 4 degrees. And then we store them in a fridge. So there are still some more filters to be analyzed in these Petri dishes. Once we are starting to analyze a Petri dish, we go to a sterile bench.
Grunz makes himself comfortable at the work desk. He wears plastic gloves and cleans the surfaces of the workspace with ethanol to keep everything nice and sterile. In front of him, in Petri dishes, are some of the filters from the glacier, ready to be processed.
On here, on this filter, are now the phages, the viruses that we filtered. So what we do basically is we cut up the filter in smaller pieces. So I would make some amount of squares.
Once the filter is cut up into small squares, Grunz puts the bits of filter into a 50 milliliter tube using a pair of tweezers.
So the next step would be to extract the virus from the filter.
Exactly. So afterwards I'm adding this buffer to this tube as well, so that all the viruses go into the solution and are not attached to the filter anymore.
That buffer is a clear liquid used to conserve microorganisms and viruses.
So I'm basically now adding this buffer.
So you said that you still don't know what kind of microorganism and virus are on the filter. So they could be also pathogen. Are you not afraid? You don't need like protection?
Well, first of all, when they're bound to iron, they're not infective, so they can't infect anything actually. And furthermore, these are just viruses from the environment, and if I go hiking in the Alps, there will be basically the same risks. And since these viruses have been like in the ice for hundreds or thousands of years and are really adapted to the cold, and there are no humans in the glacier, I guess, so it wouldn't make sense for them to have a human as a host or any bigger animal, I guess. So mostly the host of viruses in glaciers are bacteria, so they infect other bacteria, or maybe some small algae and like other microorganisms, fungi.
The plastic tubes containing the bits of filter and the buffer liquid now go into a cold chamber, a room at 4 degrees. Grunz sticks the tubes with some tape onto a rotating cylinder so the viruses and the liquid get mixed well.
And now you would just do this throughout the whole night, and then say tomorrow all of the viruses would be in the liquid part. Exactly. Basically afterwards we can use there's commercial kits that are available to then extract the DNA, and then you can really elute only the DNA in a little tube, which you then can send to this external company for sequencing.
After the sequencing, the moment has come to test what this bacteria can do.
We do genetic analysis, but we do also cultivation.
Team leader Beat Frey again.
We bring the organisms directly in cultures here in the lab, so we have living cultures, and then we can study and characterize interesting metabolic pathways of these organisms.
Biologist and scientific assistant Anja Wertz tests the bacteria against several characteristics.
So at the moment we are screening these microorganisms for plastic degradation to see whether we have some isolates that could potentially degrade plastic. And we also screen them, for example, for antibiotic production. Here is an electronic microscope. So everything you put under the microscope will be displayed on this display. And currently on the display is an antibiotic test. Here on the right side you see a bacterium, which is an isolate from the biobank.
We'll get to that biobank in just a moment.
And then on the left side there's a fungi growing. But here in the center we have an area where the fungi doesn't grow. And so this shows that this bacteria secretes some antibiotic that inhibits this fungi from growing.
The work has just begun. There are enough bacteria and fungi to be analyzed for years to come. And so they're stored in a biobank. Anja Wertz takes us to the basement. There are several deep freezers in a long corridor. She opens one of them and pulls out a stack of boxes.
So these are the bacteria coming from the glacier and the permafrost?
Yes. They're stored in little tubes inside these boxes. So if you look at it, inside of this box there are tons of these little Ziploc bags, and inside these bags there are these little tubes, and each of these tubes contains one bacterial isolate. And once we need them we can take them out and then streak them out on a nutrient plate where they grow again.
You mean that maybe here we have a bacteria that can produce some antibiotic like in the future?
Yes, it's possible. So in theory, there are some bacteria that are already known to produce a lot of different antibiotics. So we will very likely find strains or isolates that can produce antibiotics. If it's a completely new antibiotics, we don't know that yet, but we need to research that further. So it's possible that we could find a completely new kind of antibiotic.
The samples in this biobank are not only from Swiss glaciers.
We have bacteria and fungi, around 1,500 isolates. So this would be around 1,200 bacteria and around 300 fungi. They come mostly from Switzerland. Some are from permafrost floors and some are from the soil that is on top of the permafrost. And then we also have a few that come from a glacier environment. And then we have some others that are not from Switzerland, but from northern Greenland or Svalbard, so from Arctic environments. The cold environments of the high alpine regions and also the Arctic are changing drastically in the next decades because of the climate change. And when these cold environments disappear, we also have a lot of cold-adapted microorganisms that can't survive this new environment. And so that's why we're preserving them now, so that we have them for sure, and then we can research them before we completely lose these strains.
That report from the Swiss Alps came from our journalists Luigi Iorio and Celine Stegmuller. And we'll be back in the new year with a special season called Lost Cells. There we'll uncover the misconduct of a Swiss-based stem cell bank, weaving together personal accounts from families in Spain, Serbia, Italy and beyond. In the meantime, if you like our podcast, hit the subscribe button so you don't miss any future episodes. But for now, season's greetings from all of us at Swissinfo, and please join us again soon. Today's episode was recorded and edited by our science and video journalist Michele Andina. For more content, visit our website, swissinfo.ch. I'm Jo Fay. Thanks for listening.
Hello, I'm Imogen Folks from Swissinfo's Inside Geneva podcast. On February 24th, 2022, Russia attacked Ukraine. The invasion caused Europe's largest refugee crisis since World War II, and during the year-long conflict, tens of thousands, possibly hundreds of thousands of people, soldiers and civilians, have been killed. Over the past year, a number of episodes of Inside Geneva have looked at the heavy humanitarian toll of the war and its wider implications for the world. We've been joined by historians and international human rights experts to ask about the background to the invasion. We've talked to major UN aid agencies about how the war in Ukraine is impacting other humanitarian crises. And we've asked if sanctions or war crimes investigations can stop or at least limit this conflict. If you're particularly concerned by the war in Ukraine, do listen to these episodes. You can find Inside Geneva free to listen on Apple Podcasts, Spotify, Google, and all your usual podcast apps.